My project, I'm one of Dr. Alessandra Catanazzi's graduate students, and my project that I used the Tinker money for is recognition of chytrid scents by the strawberry poison frog. So John talked a little bit about what the chytrid fungus is and what it does and such, and I have a little bit of an overview here too. So just as a reminder, the chytrid fungus, there we go. The chytrid fungus arrived in countries at varying times throughout the 1900s, but it really just kind of blew up and became a big problem starting in the 1980s when people were seeing mass die-offs in Central South America, Australia, etc. And now that it is a worldwide fungus and a really big problem, there's been so many studies on the various effects of the fungus. And really what it does is it is a skin infection that, because the skin is the breathing apparatus of a frog, ultimately kills it. And so it can go anywhere from skin lesions to skin just completely being removed. Then osmotic regulation of the frog is off, which then leads to cardiac arrest. Frog is dead. And it, according to recent research, seems as though we can attribute over 100 extinctions of frogs worldwide to this fungus. Many more declines in the 500s, 600s species are declining because of the fungus. And so it's still a very big deal about 50 years after it became a big problem. So now that you know about the chytrid fungus, what do frogs do when they are infected? Well, a lot of them die, which is the problem. They can't handle the infection. They can't clear themselves of infection. It hits the immune system and they just shut down and the frog cannot survive. Some frogs can have immune resistance. And so, as John said, there are some frogs that can clear themselves of infection. And you can also handle sometimes a small load of infection. And if you make it so that the infection does not increase too much, then the frog should be okay. But the facet that I'm curious about is, what about behavioral avoidance? And so if we remember from COVID-19, when we figured out that 6 feet apart was going to help us to stop transmission between people, it became an action that we took in order to try and stay safe and keep others safe. So is there behavioral avoidance that this frog can detect and avoid the chytrid fungus? Can frogs do this in general? We really don't know. There's been only a few papers that have looked at this in a lab setting, and they've only tested 5 species. There has been proof, or has been suggestion of behavioral avoidance with toads, but there's really just a breadth of research here that we have not looked into. We don't know within this recognition and avoidance what kind of recognition it is. And so the 2 types you really think about are chemo and mechanosensory. Chemo being, I smell this smells bad, or I smell this, I recognize this as a bad smell, I'm going to stay away from it. Mechanosensory, I'm touching this is burning me, this is hurting me, I'm going to stay away from it. We don't know which type of recognition that it is. And we have no idea how this could relate to other behaviors that organisms exhibit. For example, frogs that exhibit parental care, can recognition of this fungus lead to different parental care strategies? So my study questions for this chapter were, what is the effect of chytrid scent presence on adult frog presence in caged experiments? And what is the effect of chytrid scent presence on tadpole deposition in a natural setting? And I used this frog you see on the bottom right. I took this picture in Costa Rica. Well, I'll get to that in a minute. But my study location was La Selva Research Station in Costa Rica. You can see on this map, La Selva is this greenish area, and then they have a river that feeds into it. Specifically in La Selva's area, I was in secondary forest closer to the field station on trails. And I was doing various things with cups and recognizing frogs and such while I was there, along with caged experiments. And my study species, the strawberry poison frog, Oophaga familia, also known as the blue jeans frog, this is the most famous color morph that we know of, is a very tiny, very tiny, diurnal, meaning usually works mostly during the day, by parental care species. And so the males protect the eggs once they are laid, and the female moves and feeds tadpoles once she puts them in water. And they are a terrestrial frog, similar to what John was talking about before. They don't live in water, but they need to stay moist. And so we also know that poison frogs, other species have been tested. And when you put decaying leaf litter around a water source, they will use that more often, presumably because since the leaf litter is decaying, they know that it's a stagnant water source and it's safe for their tadpoles to be put into. So it seems as though they may use scent to be able to tell if a water source is good or not. So for my 2 experiments, I needed to be able to collect Chytrid scent for both of them because I'm looking at chemosensory. I was not allowed to take the physical fungus to Costa Rica, which makes sense, and so we had to figure out another way of catching it. And Dieta Mills at Florida International University gave us the idea of catching on gauze strips. What you do when you're trying to teach dogs to recognize recognize COVID and then sniff it out from people is you collect it on pieces of gauze, the stuff that you put into wounds to pack it and such. It is really great at collecting scents. So I had mason jars and I put the Chytrid in the bottom. I had 2 different types. You had Chytrid in broth and you had just the broth, which I use as a control. Put a bunch of gauze strips on the top, make sure that they don't touch the Chytrid liquid in the bottom, and put it at 37 degrees Celsius for at least 24 hours in order to collect the scent. And then I put that into individual bags and then into aluminum foil bags to make sure that the scent was taken. And I did all of this before I went to Costa Rica, so I had to catch all of this. So I think the video is playing. You're seeing a sped up version of, and you can see John in the background because he helped me with this. And this was like a 30-minute period of us just putting scent things into bags. I had thousands of these little bags that I took with me to Costa Rica. It was insane. So this was the collection process before I went to Costa Rica. Once I got there, for my first question, a reminder is that this is where adults will stay in caged experiments when I give Chytrid scent in certain areas. Catch frog pairs and I place them in experimental cages. So we measured them, we took photos because they have spot patterns so that you can identify them for really well. And I put them into sets of cages and I had 12 sets, so 24 frogs total, 12 pairs. Each of my containers was 2 butterfly cages connected with a piece of plastic. And when I put the frogs in, I put them in that connecting plastic and they got to choose where they wanted to be. And then I watched over time where they wanted to be over the time that I did my experiment. So 2 connected sides. In one side, I was going to have the smell of the control, the broth. On the other side was the smell of the Chytrid. And within each side, on the right, you can see you had leaf litter and 4 of these little cups, which they would use to deposit their tadpoles if they chose to breed. And you can see a pair appear, the male's trying to call out the female. And if I remember correctly, she was having none of it. My daily data collection for this was I acclimated the frog pairs for 8 days. And then I added scents for 19 days total. I looked at daily presence. I was checking morning and evenings for a while and then just checked evenings. And so I have measurements of 9 mornings and 18 evenings. And I randomly assigned for each cage which side the scent was going to be on. Sprayed each day with rainwater, replaced gauze every 4 days. And so let it sit in there for 3 days. On the 4th day, replaced it, fed the frogs, and ultimately they were released after this experiment was over. Results-wise, I looked at side preference. So do you care which side of your cage you're on? Do you care where the scent of each thing is on? And do pairs tend to stay together? Tested normality, equal variance. It was non-normal data and unequal variance. And so I used Wilcoxon paired t-tests because of that. And I did observe, although I didn't really look at the data on this, but it did seem that when a female would move which side she was on, in a couple of days or even 1 day later, the male would follow her. So it did seem as though the female was choosing places and the male was following her. So it might not be that the male's choosing necessarily because of one of these variables. It could be that he's choosing based on where the female is. Data. They did not care before addition of scents where they were on the left or right side. They did not care in the morning after I started putting scents in. They did not care in the evening. So nothing was significant with sides. So one side did not entice frogs more than the other one. When I started putting scent in, they did not care in the morning. They did not care in the evening. So it seems as though the scent of the fungus did not tell them which side they wanted to be on. But as I said earlier, frog pair sides, males seem to be following females. Before addition of the scents, they were spending more or less equal time apart and together. After, they were spending much more time together. That could have been a time thing, or that could have been various factors that I would need to do extra experiments to figure out. So side and Chytrid scent don't matter. Likely over time, pairs intend to spend more time together. It could be a time thing, but I'd need to do separate experiments to figure that out specifically. So that's first experiment. Second one, I hung cups up on the trails in La Selva to try and get frogs in the natural setting to deposit their tadpoles. And so you put spoons up, zip ties on the spoons, medicine cups hanging there, and each medicine cup was doubled up, and I poked holes in it so that water would not completely overflow in case a tadpole was put in. You didn't want to wash the tadpole out. And then we doubled up the cups because people were thinking that it might be that they wouldn't want to deposit their tadpoles if they saw like a piece of gauze in there. And so the gauze was in the bottom, kind of sort of hidden, and then they would be able to deposit the tadpoles in the top. I put cups on 154 trees. I had 308 total cups on 3 different trails, 50 to 54 trees per trail. I have, again, another video of setup, and you can see on the left-hand side, this is what each setup looked like, not with the cups doubled up yet, but you had A and B, and I randomly assigned which side was going to have the control scent and which one was going to have the Chytrid scent. Filled it with rainwater, made sure each day that if it had washed out too much water, I put in some extra water, etc. Setup. Cup setup and acclimation, I did that from July 7th through July 24th, and then I added scent for 20 days. Checked every day for tadpole deposition, replaced rainwater as needed, and replaced the gauze every 4 days. My results, I only had 2 depositions total. And for people who've used this before, they've gotten like 40 depositions in a month. And I was there for a month and a half, or actually, they've gotten about 200 in a good month. So I was expecting at least 10, and it did not happen because rain was weird that summer. So unfortunately, I can't really say much about this data because I don't know much. One was deposited before scent was put into the right side of the cup, as you can see in this picture here. The other one was deposited after scent started to be put in, and they were put on the Chytrid side, but I can't run any stats on this. So conclusions for this, it seemed as though where I put my cups it was closer to trails. They weren't used too much, but it could have been that it was a little bit more disturbed than these frogs would like to start using the cups. And so moving them somewhere else might have been better. Tentatively, location doesn't matter for where the smell is. I can't say that for sure. I'd need to do more data collection. So future directions, reminding for the first experiment for the sides of the cage experiments, side and Chytrid scent location don't matter. I need to test the gauze method, make sure that it actually captures volatiles properly. And I'm doing lab chemosensory tests now. They're supposed to be done before I went into the field. It did not happen. And so I'm kind of doing a post thing to see if they even care about scent of Chytrid at all. Over time, it seems pairs intend to spend more time together. So I think that the experiment being longer would have been well. For my second one, previous success, more off trail. I moved cups in case I was able to go back to Costa Rica or needed for additional data collection. So I have 600 cups out there currently in case I need to go back and do more. And I have it in an off trail location. And need more gauze additions and a longer time for this to see if I get any more depositions in cups. If my lab results are positive, I would say it might warrant an additional field season to see if there's anything there. But if the frogs don't care about the smell of Chytrid, then I don't necessarily see another field season being warranted. Finally, acknowledgements. So many people to thank. Tinker, FIU, I got a La Selva grant, and through the Organización para Estudios Tropicales, for money to stay there. I had amazing staff, Danilo, Enrique, Orlando, and everyone who took care of us at the field station was amazing. And I went to the field by myself, and people were looking at me and going, why are you here alone? And so I had so many people who helped out. Alondra, Andreas, Chloe, Tony, Chanel, and Cullen were amazing. And my advisors and committee members and lab students, fellow graduate students, and Dieta Mills for the gauze thought for collecting the fungus. So with that, if I have time for questions, I was not following my time, so I don't know where I'm at. But thank you very much for listening.
Likewise, if you have questions, please ask them over there. I'm gonna ask you a question, Rachel.
Okay, thanks, Alessandro.
What are you doing now?
I am currently in a lab. I got frogs from Bocas del Toro in Panama, and I have them in a lab here, and I'm doing scent stuff with scent chemosensory chambers to see if the frogs care about the scent of Chytrid. And I'm doing mate choice experiments. I'll be infecting a set of frogs next week to see if a female cares if a potential mate is infected and if she would choose to stay by the one that is not infected. And I'm also doing dorsal brightness photos to see over time with infection if color or intensity of brightness on the back of the frog changes, and alkaloid stuff because these frogs have toxins, seeing if that affects Chytrid. I'm doing a bunch of different things here.
Great. Thank you. All right. Thank you.